Today we're going to be working with marine sedimentation. We're going to be looking at different sources of sediment from 3 different beaches. We're going to look at a sample from Newport, California. We're going to look at a sample from Newport, Oregon. And we're going to look at a sample from Bermuda Island. What we're going to do is we're going to just take a pinch of sand and a pinch of sand at random. And we're just going to place it in a shallow dish. Then we're going to use our Microscope, turn the light on, and using the handheld mode, we're going to zoom in just to any particular place on that scope and snap away. In this particular picture, we don't have any shells or any signs of any biologic resources, nothing living. These are all sand samples. This particular sand sample is from Newport, Oregon. As we look at our sample, we can see several white and see-through structures, translucent. We can see definite shades in our yellows. We can go to darker oranges. We can go to browns and even blacks. What we're going to have to do is learn how to identify these particular minerals. Most of the white ones that you see are quartz. Okay? Most of the dark ones that we see are basaltic. So we're going to be looking at a comparison between 2 totally different types of sources. What I'm going to do now is take another snapshot, but this time more into this area. And what we have now are biologics. So what we have here are different shell samples, different fragments where shells have broken apart. And what we're going to look at more closely, and we'll switch lenses to look at, is exactly what kinds of shells these are and any other parts that we might find. For example, here we have a nice hydroid colony that's attached to that particular shell, and we'll be able to look at it and also each of the zooids, each of the little animals that it's made up of. Secondly, we'll switch over to another sample. This particular sample is our sample from Bermuda. And I'm just going to zoom in here and take a picture. And what we have in this picture is a beautiful sea urchin spine. We also have all kinds of shell fragments, and notice, no sands at all. Okay? We have some coral fragments also, and so we'll take a better look at this particular sample, and we'll be able to compare it from an island sample to basically a terrigenous beach sample. The third sample we'll be looking at is a sample from Newport, California. This is in the Back Bay, so I'm going to take a couple of pictures. And our first picture this is going to be interesting in comparison because what we're looking at is fragments, just little tiny pieces of dust. Okay? The Back Bay is further back inland, and therefore the wave action has to bring the sediment source in further. Also in this area we're going to look at some marine sediments, and I think I can get some nice shells in here. And there we go. So let's look at this sample. And as you can see, there's a nice huge shell sample here. There's another shell sample there and one down in there. And again, when we look at with a higher power lens, we'll be able to tell exactly what types of shell these are and what organisms they come from. Now what I want you guys to do is I want you to again take any sample that you want. Again, start with a 50 power lens. Look through the sample and capture several images from each sample. Capture terrigenous sediment samples, if there are some, and then also look for some of the biologic samples and capture them. What we're going to do is use the handheld scope, and the reason why is because it will allow you more flexibility in movement, and you'll be able to see things from the top. You may also want to go and use the microscope on the stage, and this way you can put your sample on top. You can get a lit example from underneath, which will allow you a back shadow effect, and you'll be able to see shapes of the sediment sources better. Second part of this lab is what we're going to do is take a random sample of just sediment no biologics. And what I want you to do, very carefully and slowly, in a petri dish, is count out 100 grains. The scope, of course, on low power, will help you look through those grains. And after you've counted out 100 grains, then what I want you to do is sort them. Take all of the clear glass ones and make a nice little neat pile of them. Take all the black ones and make a little pile of them. Take any red ones that you see and make a pile of them. When you get through sorting, what that will automatically do is give you percentages, since we're based on 100. So if you have 37 white clear grains, 37% of your sample is quartz. If you have 7 dark black grains, then you have 7% of your sample being basaltic. As you take samples up and down the coastlines of California, through Oregon, through Washington, you can literally see those percentages change as you go from more of a granitic type beach to more of a basaltic type beach. After we graph all of these things, we will notice that there are some different looking graphs. Each of these graphs is actually termed a beach profile. And as we look at each beach, we can compare their profiles to determine sediment sources, ages, and even the types of sediments that are found on the beach.
Yeah, is that a shell or is that a rock?
The sediment lab is a fairly lengthy lab. The kids have to realize what sediments are, what minerals are, in order to describe their sediment samples. Part of sediment, and not on every beach by any means, are biological material too. The pieces of shell, the pieces of crab, a fish scale, a fish tooth, a piece of dried up algae, whatever happens to get washed up with the last wave is going to be part of that sediment. That tells me about what lives off of that beach, and that's an extremely important part of that sediment also.
This looks like a piece of shell.
Yes, it is.
Mr. Schmidt,
Yes.
can you tell which species of sea urchin it is just by looking at the
Alex,
spine?
look at this.
Sometimes you can. Every beach has a fingerprint. Just like each of us are different, and we can identify ourselves by our fingerprints, beaches have fingerprints also. I know when my students get through with this lab and they go down the beach, they look at that beach differently the next time. And they come back and tell me that. And this is where the teacher comes in, giving students opportunities. Opportunities to look, opportunities to learn, opportunities to explore new things they've never looked before. The microscope gives us a tool that allows us to literally look at structure. Sands have different shapes to them. And by looking at the structure, we can tell relative age of a beach. For example, if the sand grains, and we have to use a microscope to see this, if the sand grains are very angular, that means they've just been broken off of the larger minerals. That means the sands haven't traveled as far, nor have they been washed with wave action as long. We have a choice of using the handheld microscope in 3 different fashions. We can hold it by hand. We can mount it on a tripod or on a device that will hold it for us. And we can turn it upside down and put a flat stage on it. And each one of those becomes a different tool. Okay? So if we wanted to look at a prepared sample, we're going to use the stage. If we need to use both of our hands for manipulation and pulling, then we need to put it on a tripod so we'll hold it for us. And if we want total flexibility and we can manipulate it, then we want to hold it in one hand and hold our object that we're looking at in the other hand and really get down and dirty and get into there, okay? In the sediment lab, you are basically going to use it handheld. The reason why is you want a top view of the sample. You need a good light source, okay? And you basically want to look at color. You want to look at texture. Is it smooth? Is it dimpled? Is it rough? Those tell us something. Okay? Is it dull? Is it shiny? Is it translucent? We'll be able to see those factors with our handheld microscope looking directly at it. We also want to reverse that. We want to take our stage, we want to put a sample in a clear plastic small petri dish, and we want to backlight it. And that way we'll be able to see silhouette And by looking at the silhouette, we'll be able to tell the roundness and the sharpness of the grains. A microscope is just another tool. It's another resource. Just like the computer, just like your textbook, microscope allows us to expand our vision. The more resources you bring into your classroom, the more successful you will be at teaching that topic. Handheld allows you flexibility. It allows you to move around. It allows the students to be a little bit more creative. What I have to do is bring those critical thinking skills out. I have to show them a comparison. I have to make them use their minds to describe back to me how things are different and how things are the same. The digital microscope allows them to see that,